Hey guys, welcome back. This is going to be our last video, our last set of notes here for chapter 7. And we're going to cover a little bit of ground this time. I've been breaking it into little chunks for you so far, but we're going to go ahead and power through here. We've spent all of our previous videos talking about, kind of the ideal energy production conditions. You've got mitochondria, and we've got oxygen, and we've got glucose, and we've got all these things we need to make energy. We end up making lots of ATP. Well, today we're going to look at some places where that's not the case. There's a few organisms that live in a situation where mitochondria, oxygen aren't available, but they've got to stay alive. And we're going to talk about some ways that they accomplish that. Pardon me. So this section is called fermentation. Fermentation is an energy production pathway that still starts with glycolysis. We still have that first stage where it's still in the cytoplasm, and you still have glucose, and you still make pyruvate, you still make NADH, you still net 2 ATP overall. Okay, not a lot of energy, but it's some. So now in fermentation, our pyruvate is going to be converted to something different, like anaerobic respiration, but unlike anaerobic respiration, we will not completely break it down to carbon dioxide and water. That's ideal. That's what we do in mitochondria. Fermentation guys, they're not going to get this job done. So their goal is to regenerate NAD+. NAD+ is the reactant in glycolysis. And so if all you can do is make 2 ATP at a time and you want to stay alive, you want a lot of this guy because he's your starting point. So we're not going to produce a lot of ATP. We're not really going to produce any ATP once that pyruvate is made. Our goal is to take him and get him back up here where he's useful again. And so it's going to provide about enough energy for some single-celled anaerobic species. No oxygen, they're really small, they're not moving a lot. We're talking about yeast, some bacteria, those kinds of guys. All right, so there's two types of fermentation. There's alcohol fermentation and there's lactic acid fermentation, and we're going to talk about them both. So our first example of fermentation is alcoholic fermentation. Alcoholic fermentation, we go through glycolysis. We make our pyruvate. There's no oxygen. We go to fermentation. Pyruvate gets split into acetaldehyde and carbon dioxide. This acetaldehyde compound is going to get some electrons and some hydrogen, and it's going to form NAD+, and it's going to form ethanol. And so if you were asked, where or what are our products of alcohol fermentation? Your answer, NAD+ and ethanol. Where does it happen? Cytoplasm. Okay, single-celled organisms like fungi are how we're going to get this job done, and we will use alcohol fermentation to make things like beer, wine, bread. Lots of food products are made by fermentation, and the fungi are kind of weird to think about, these little living organisms that we use to make bread, but that is what we use to make bread, and alcoholic beverages are these little fungi. So kind of cool. So here's your cartoon of it. Glucose, just like we talked about before. Glycolysis, just like we talked about before. We put 2 ATP in, we get 4 ATP out. We've got 2 net gain, we make pyruvate, and then things change. We lose some carbon dioxide, we make acetaldehyde, we make NAD+, and we end up regenerating that NAD+. So the sad thing, I don't know if it's sad or not, the bad thing about it is we produce these 2 NADH, which I told you, that's good news, they're the energy nickel. And then in this process, we use them right away. So they're not the energy nickel. They're not being used for anything later because there is no later. There's not mitochondria. There's not a big old ATP synthase waiting to make a bunch of energy. We have to settle for regenerating NAD+ and bringing it back to this step to just make these 2 into 4. And so it's not a lot of energy. That's the bottom line, but it's enough to keep a little cell alive. Now there's another one, lactic acid fermentation. You and I don't undergo alcoholic fermentation when we run out of oxygen, which is good because you'd go for a long run, get really tired, and be drunk at the end of your run. That would be horrible, right? We go through lactic acid fermentation instead of alcoholic fermentation, but it's similar. You still go through glycolysis, you still make pyruvates. Your pyruvates don't have time to go through that Krebs cycle and make all those ATP in the mitochondria. You just need energy fast, The pyruvate will actually go through a similar process to make NAD plus and lactate. And so our products here are lactate and NAD plus. And this is carried out by beneficial bacteria that we kind of throw under this general term of lactic acid, but they're carried out by these beneficial bacteria that we use to make yogurt. So you get a lot of probiotics from yogurt, good bacteria in yogurts, and we use them in this fermentation process of making yogurt. So it's not just in bacteria, it's also in animal skeletal muscle cells, particularly the white ones. White muscle cells, that is, not white animals. So white muscle cells, there's red muscle cells as well. Red muscle cells, long distance kind of stuff. White muscle cells, sprinting, lots of energy for a short amount of time. So the cool thing about it, it'll give us a lot of energy, but there's a cost, and that cost is making a bunch of lactic acid in our muscles. It's the reason you're sore or muscles burn when you sprint, right? So it's useful if it's quick, strenuous activities. Weightlifting, that's quick and strenuous, You're only lifting for, 30 seconds or a minute for one exercise before you change. But during that quick, strenuous activity, we need a lot of energy fast, and we can make lactic acid during that. And it does not support prolonged activity. It's only good for short bursts of energy. But that's lactic acid fermentation. It's the reason your muscles are burning and sore when you're done working out. So here's the cartoon of it. We still start with glucose. We still start with sugar. 2 ATP still go in for glycolysis. 4 ATP come out. We still make pyruvates, but instead we convert them into lactates. We quickly take the NADH that we made, make it into NAD+, get that NAD+ back here so we can make ATP again. So very similar. Really similar to the alcoholic fermentation. It's the end product that's different. All right, so we talked about all of these ways to make energy, but they all started with glucose, right? Not everything you and I eat is sugar. A lot of what we eat is carbs, but those carbs are broken down into glucose, and glucose is the beginning of glycolysis. But what about stuff like fats and proteins that we're eating that aren't carb-based? We still get energy from those. So what's the deal? Is it just glucose, or do these guys have a job too? And the answer is they, of course, have a job too. We still get ATP from fats and proteins. And so just like metabolizing glucose, which has been our focus lately, we use lots of coenzymes when we're digesting these fats and proteins. And the energy of these coenzymes can ultimately help drive that ATP synthesis from digesting the glucose. I'll show you a couple examples of that. So enzymes, if you eat a big starchy food, bread, rice, that kind of stuff, a cookie, a pretzel, whatever, it'll take those complex carbs and start breaking them apart, these enzymes will, until we're down to little teeny tiny sugars like glucose. And then we take the glucose into the cells, we turn it into glucose 6-phosphate, and it's glycolysis, and it's acetyl-CoA formation, and it's the Krebs cycle, and it's electron transfer phosphorylation, and the whole thing we just learned. Now, what if you've eaten enough sugar, which we're in America, let's assume we have, that you've got enough glucose in your cells and we don't need any more? What if you eat more sugar than you need? What do? Well, we got plenty of ATP, we got plenty of glucose, you're sitting in class, you're not exercising, you're listening to a lecture on your computer with your headphones in. Here's what we're going to do with all that energy. We're going to take all that extra glucose and we're going to divert it, and we are going to build a very cool chemical called glycogen. Sorry, I tried to circle the wrong word there. Glycogen, right? We're not going to use glycolysis right now. We're going to go make glycogen. And glycogen is for people, for us, it's a long-term energy storage of that sugar, right? Plants make starch in potatoes and carrots. You and I make glycogen in liver and muscles, because there will come a time before lunch when you start to get hungry, but you're not allowed to eat in school. So you're faced with the moral dilemma of, do I sneak a tiny bag of goldfish into class, or do I let my body break down the glycogen stored from this morning's breakfast and continue to feed glucose into the production of ATP? Most kids opt for the glycogen, but I do have a few who are trying to sneak in the bags of food. So there you have it. Glucose, it's pretty simple. It's a carbon ring. You hook a glucose to a glucose to a glucose to a glucose to a glucose. You get starch, a complex carb, or glycogen, or cellulose, or something big and energetic and useful. So what about fat? We got carbs. We get carbs. We live in America. We get carbs, right? What about fat? Enzymes are going to take fats, and fats are made of a glycerol head and fatty acid tails. And we're going to take that and we're going to break those apart. And that glycerol will actually use him in glycolysis. The fatty acids, we'll convert that to acetyl-CoA and plug those into the Krebs cycle. And so it's kind of cool to see, phosphoglyceraldehyde comes from glycerol from the fat I eat, and acetyl coenzyme A comes from fatty acid chains from the fat I eat. So everything has a place that it's going to go be used in. And so the fatty acid breakdown, because there's so many high energy bonds between the carbons and hydrogens, will yield more energy per carbon than carbohydrates will. And you guys already know that. 4 calories per gram for proteins and carbs and 9 calories per gram for fat. So it's probably not that surprising for you guys. Now, if we have plenty of glucose in our blood and we're making plenty of energy and we're eating plenty of fat, acetyl-CoA is diverted from Krebs, which means we don't need acetyl-CoA in the Krebs cycle right now. We've got plenty of energy. We'll go back and build our own fatty acids and store up fat in our bodies for later use. If we're hungry, if we're starving, we can break down those fats that we've stored up in times of abundance, break them down into acetyl-CoA, and drive that Krebs cycle to literally digest some of our body to feed the ATP production that keeps us alive. So kind of a cool thing, we're able to tie in all these different food groups. Here's a fat. It's a glycerol head. It's not that exciting. Few carbons. Fatty acid tails, just energy for days with these carbon and hydrogen bonds. And that's what we break apart to turn this into glyceraldehyde, phosphoglyceraldehyde, and to turn these guys into acetyl-CoA. So proteins. What about proteins, right? Proteins are made up of amino acids. So what do we do with amino acids? Well, we can use those amino acid monomers to build our own proteins, muscles and collagen and actin and myosin and hemoglobin, and the list of proteins just goes on forever. Or we can build other molecules. A lot of our proteins, when we break them down, we will remove the amino group from the amino acid, and that's the NH2, and we'll convert them into NH3, and that's ammonia. You cannot keep ammonia in your body. That's not good, right? We don't want the same stuff that can be used to clean a swimming pool in our blood. Not okay. And so we're going to go ahead and get rid of that in urine. And a lot of the smell of urine actually comes from the presence of ammonia. Now, we can take acetyl-CoA, we can take pyruvate, or we can take some Krebs cycle intermediates, and we can actually build those out of amino acids, depending on which of the 20 we're dealing with. So we can make a lot of intermediates for ATP synthesis from amino acids. They work in these cycles too. And here's an example of an amino acid, alanine. He's 3 carbons long, and we can actually convert him into pyruvate, the product of glycolysis. We can skip glycolysis and end up with pyruvate and throw him straight to acetyl-CoA formation, because here he sits with his 3 carbons. So that's not a big jump, from alanine to pyruvate. So we can use him right away for glucose production, which is a really cool trick. So here's kind of the flow chart, not something you'd need to memorize necessarily, but just to kind of get that big picture of how everything you're eating is useful for this process of building ATP. Fats, we can get the fatty acids to acetyl-CoA. Those belong in the Krebs cycle. We can take glycerol and make phosphoglyceraldehyde. He goes into glycolysis. We can take our complex carbs that we're eating in the morning, our bagel and our toast and our fruity pebbles or whatever we're eating, and break them down into glucose and plug those straight into glycolysis. And we can take proteins and we can take those individual amino acids, and depending on what they are, we can break them into pyruvate, we can turn them into acetyl-CoA and plug them into the Krebs cycle, or we can turn them into one of those intermediates and throw them in the Krebs cycle somewhere else. And so all of these work together for that end game of electron transfer phosphorylation and the production of ATP that keeps us alive. So kind of a cool glimpse of a really big system with so many moving parts that all depend on one another and that have to work in a particular fashion. And everything that we're eating and digesting can all play a role in that. And when we have too much of something, we can store it up for later. And it's just a cool system, you know? So there you have it. End of the chapter. Man, we made it through. I'd say one of the toughest ones we're going to face this year is our metabolism, man. That's a tough chapter. Points to ponder. Why are so many diseases attributed to defective mitochondria? There's a little piece in your book that talks about that. What is metabolic water? It's a great question. You'll hear some of these weird animals on documentaries like kangaroo rat. You know, they'll say, oh, he rarely drinks water. He survives on metabolic water. So from this chapter, hopefully you understand what that means. And then finally, what happens to lactate? If we produce a bunch of lactate or lactic acid during our intense muscle activity, you know, lifting, sprinting, why is it important that we break that lactate down quickly and not just let our muscles be full of lactic acid? Those are all good questions. So I hope you understood this. I hope you gained something from it. And if nothing, I hope you appreciate kind of just how crazy the stuff going on inside our bodies is. Just a little piece of it. So thanks for your time, and I appreciate you viewing the videos.